Thank you very much. Well, I'd like to start, of course, by thanking the Molecular Frontiers Committee, particularly Bengt and Shuguang, for giving me an opportunity to return to this vibrant place. The first time I came to Hong Kong was in 1992, when I imagine many of you weren't even born. It's only after hearing the earlier talks today that I realized that what I'm going to be attempting to do is to draw together many of the strands that we've heard about to give some kind of a picture of how living things and how we, in particular, how we work. Now, ironically, I am probably the speaker least qualified to do so, but there may be a silver lining in that, because the fact that I'm not an expert might mean that I ask the kind of questions and look for the kinds of answers that those of you who are also not yet experts might ask and look for. Well, you see, here's the dream. Those of us from the UK who drove a car in the 1970s and 1980s will probably remember the Haynes instruction manuals, which were instruction books where we went to look up what to do when something went wrong with these machines. We went to look up how to fix them. Wouldn't it be wonderful if we had something similar to that for the human? A manual where we could go whenever anything went wrong with the human, like a viral infection or an allergy or a heart problem, where we could just look up the right fix. But the truth is that after pondering for thousands of years, really, the question of how life works, we still only have a sketchy idea of the answer. When I got back from the banquet last night, I should have gone straight to bed, but there was this paper that I came across in my emails that I had to read straight away by the physicist and bioengineer Stephen Quake, who said this in the paper. It's a paper in the prestigious journal Cell, and Stephen said, while all of the genes and the proteins have in some sense been worked out, we still don't understand how they function together to create the smallest indivisible unit of life, the cell. And I believe that the answer to that question has changed rather profoundly in the past few decades, thanks to the latest advances in cell and molecular biology. In some ways, the answer seems to have got only more complicated, but I hope to persuade you that it's also a richer and more satisfying answer that we're starting to glimpse than the one that we tend to hear about at school and in the media. So let me sort of start with the old picture. In the old picture, it's all about genes. You know, there's this idea that who we are, the way we are, is in our genes. So, you know, you've seen this many times. So here it is. This is what holds the genes, the double helical molecule of DNA, these two strands winding around each other, each strand containing information encoded in the form of this sequence of chemical building blocks, of which there are 4 types, which are said to act like a code, very much like the strips of magnetic tape that held binary information back in the days of the first digital computers. So each of our genes is a short section of this code, and we have many thousands of these genes that for us humans is a string that is 3 billion of these chemical letters long, and which we call the genome. So when this structure was discovered, as we heard about yesterday, in the early 1950s by Francis Crick and James Watson, it was widely viewed as the secret of life, the key to how we work. And that's what Greg Winter told us yesterday, that this is, for biologists, this is the secret of life. So the idea was that the single fertilized egg from which we all grow comes with a set of instructions in its genome, and all that needs to happen is for those instructions to be read out and thereby for us to be built. And so it seemed that once we had this knowledge, all we had to do was to read those instructions. And that was what was done in the Human Genome Project, which began in 1990 and was completed, at least as a rough first draft, by the turn of the millennium. And so here it is, our instruction booklet. So we're told this is actually a physical book, or actually 109 volumes of books, where the human genome is printed out in its entirety. And this collection is held at the Wellcome Collection in London. And it's where it's advertised as the Book of Life. But some people have questioned whether the blueprint or the instruction book is the right metaphor for the genome. A different metaphor has been proposed by the Oxford physiologist Dennis Noble, who suggested that a better analogy might be to a musical score. And that's certainly better in some respects. When we listen, let's say, to Beethoven's Third Symphony, to the Eroica, to what can we ascribe the cause of the music? Now, we might say that it's the score that Beethoven wrote, but you can sit there for as long as you like looking at the score and you will not hear any music at all. Well, that's obvious, right? Obviously, in order to produce the actual music, you need an orchestra to play the score. Similarly, put a human genome, the entire genome, all our chromosomes, into a test tube, and it will just sit there and never do anything. You can throw at it all the ingredients that we might think it will need to do something, amino acids and lipids and all the rest of it, and still, it won't do anything. It won't produce a single cell, let alone us. The only way you can get life out of a genome is to put it in something that is already alive. So the problem with saying that the genome is the secret of life is that evidently the genome is not what makes life happen, not alone. Sure, life won't happen without one, just as Beethoven's Third Symphony won't happen without an orchestra or without a score. But what we're really saying is that for life to happen, the genome needs to go into an already living system, at the very least into a single cell. And in the picture that we're given of how we and other organisms work, what lies between the genes and the organism is often presented as just a black box. And it's sometimes said that what happens inside that black box is so horrendously complicated that it's best that you don't even look. But just rest assured that scientists, like the brilliant scientists here today, are working on it, and that one day we'll have it all straightened out. The problem is this, though, that there's no life in the genes, and so clearly this is where life happens. And so to understand how life works, we really do need to take a peek into that black box. And what is inside it can look mind-bogglingly complicated, but I hope to persuade you that we can start to make some sense of it. And what's more, that over the 2 decades or so since the human genome was completed, the sense that we have started to make tells a rather different story from the one that we've traditionally been told in the genomic era. So following Crick and Watson's discovery of the structure of DNA, it was found that what the genes in DNA really seem to encode is instructions for making proteins, the molecules that catalyze the cell's biochemical reactions. So here is Greg's other answer for the secret of life, the one that chemists like, which is it must be all about enzymes. And this decoding, as we've heard already, happens in 2 stages. First of all, the information in a gene is copied from DNA into a similar molecule, this messenger RNA molecule, and then that is seized by this molecular machinery called the ribosome and translated into the string of amino acids that then folds up into a protein. And there are many thousands of these proteins in every one of our cells. There was a picture a bit like this shown yesterday. This is actually, although it's a computer-generated image, it's actually based on real experimental data for what the inside of one of our cells looks like. And you can see that it looks horrendously complicated. But that doesn't matter, the usual story goes, because each protein has a very particular shape, which fits together like a lock and key with the molecule that it's meant to transform. And so it will only recognize and work on that particular molecule, and it will ignore all the others. And so through this series of highly specific molecular interactions, proteins somehow put together us, or more generally, what's called the organism's phenotype, what it looks like and how it behaves. And this idea is commonly expressed as what Francis Crick called the central dogma of molecular biology. And Bob Langer showed us a version of this yesterday. This is actually Crick's original version of it, where he was cautious enough to, with these dotted arrows, to suggest that maybe sometimes the information can sort of go other ways. But the key point is that the idea is that the sequence information is going from DNA to RNA to protein, and once it's got to protein, it's not going to go back again. So the idea is that we've got our genotype in DNA, and then somehow, once we've got proteins, they put together the phenotype, the organism. Since the Human Genome Project, more and more complications with this sort of picture have emerged, and I want to tell you about some of those. So I said we have thousands of genes making thousands of different proteins. How many exactly? Well, a banana has 36,000 genes. And I don't know about you, but I like to think that I'm probably more complex than a banana, and so I'm probably going to need more genes than that. And when the Human Genome Project started, a common estimate that scientists would give for the number of human genes was that it was probably going to be in the region of, maybe I can move it on here. Maybe I'll point it that way. Yeah, there we go. Probably in the region of 80 to 100,000. So pretty quickly, once we started to get actual data, this was shown to be a considerable overestimate, and it's now thought, so this is once we started getting the data, the dotted lines are the estimates, the solid line is the data, and you see it's far less than that. It's about 20,000 genes, possibly even a few fewer, maybe as few as 19,000 genes. That's as many as a tiny soil-dwelling worm called the nematode, and it's scarcely more than half of what a banana has. Now, these numbers are often sort of presented now as an example of how wrong expert opinion can be. But I think the important question to ask is why they were wrong. Did we perhaps have the wrong idea about exactly what role genes were playing in putting us together? Well, here's another complication with this picture. In the 1990s, there were one or two genes known for which the biologists studying them didn't seem able to find the corresponding proteins. And in the end, they had to conclude that this is because there aren't any. That those genes simply encode RNA as an end in itself, RNA that has some biochemical function that doesn't just act like an intermediary, like the messenger RNA, but that does the kinds of things that proteins do. And because this RNA doesn't actually encode proteins, it is called non-coding RNA. So there were a few of these non-coding genes. Well, okay, biology is full of weird exceptions, but this wasn't an exception, because over the past 2 decades or so, the number of these non-coding genes has kept creeping up. And a few years back, it seemed, by some counts at least, to overtake the number of coding genes, genes that encode proteins. And what's more, some current estimates are that there are going to continue to be lots more of these non-coding genes found, perhaps so that they will turn out to vastly outnumber protein coding genes. So you can see that the genome isn't really quite about what we thought it was all about, which was making proteins. It's partly that, but not entirely that, and perhaps even not primarily that. And yet somehow we still seem to be telling much the same story that we were telling about it as we were back in the 1990s, or even the 1970s. That doesn't seem right. Well, it's even more complicated than this. The old idea, and we heard about this yesterday, was that the information in a gene encodes the shape or the structure of the corresponding protein. And it's that shape that lets the protein do its job. And here's one that we've seen several times already. This is hemoglobin, and it has this very characteristic shape. In the analogy of the genome with a musical score, we might say that it's the score that prevents the orchestra from making just a random noise, because the score tells each player exactly which notes to play and when. And that's the equivalent of how a protein's gene-encoded shape, like this one, tells it what to do in the cell, which molecules to grab hold of and which to ignore. So it's that lock and key aspect that I mentioned earlier. But we now know that for many of our proteins, including some that have the most important jobs in our cells, the DNA score isn't really like this at all. It's left open to interpretation. And what I mean by that is that some genes encoding some proteins don't assign it a well-defined structure like this. The structure is left rather loose and floppy, or as biochemists say, it's intrinsically disordered. And Alan Furst mentioned briefly this class of disordered, intrinsically disordered proteins. Now, this isn't some failure of the genome to give these proteins a proper shape. It's a deliberate feature that evolution seems to have, so to speak, chosen, because there's actually much less intrinsic disorder among the proteins of simpler organisms, like bacteria, than there is for us. So it seems that evolution has found disorder in proteins to be something useful, and perhaps even something necessary for making complex, multicellular, multi-tissue organisms like us. Now, perhaps a third or maybe a half of all of our proteins have parts or wholes that are disordered, including, as I say, some of the most important. I mean, the protein P53 was mentioned yesterday, one that is a crucial gene and crucial protein in many cancers. So that is one of these disordered proteins. It might seem odd that we didn't really know about the importance of protein disorder until a couple of decades ago, but that's because the methods that were generally used back then to look at protein structure really only worked for proteins that were ordered and which could therefore be made to form good crystals. And it's a reminder that in science, and probably in life really, we tend to see the things we expect to see, and to study the things that we have techniques for studying, rather than necessarily studying what is there. Proteins with intrinsic disorder like this tend to be far less choosy about which other molecules they stick to. They're rather indiscriminately sticky, which is to say they're promiscuous in their molecular unions. So P53, for example, the P53 protein will bind to various other proteins and to DNA. So, it was said that, and it quite rightly said, that P53 is a tumor suppressor, but it doesn't necessarily do that business in just one single way. It has many partners that it binds to, and it's also involved in other processes in the cell, like cell senescence. So the old idea that the molecular chaos of the cell is kept orderly because each of the proteins is very selective about what it interacts with, you can see that doesn't always quite work. Now, the standard argument for how we get to be so complex with so few protein-coding genes goes a bit like this. The idea is that it arises from the complexity of the network of interactions between proteins. So this horrendously, this is the kind of picture that you can see every week, really, in nature and science, where there are these complex, networks of interactions between many different molecules. And one of the consequences of these interactions and these feedbacks is that you can get proteins made by one gene that can affect how whether another gene is on or off, or how much its protein is produced. And this notion of some proteins switching on or off other genes, or in general, how genes are switched on or off. This is the business of gene regulation, which we've heard a little bit about already. Now, this isn't a new idea by any means. We knew about gene regulation at least since the 1960s, when the French biologists Jacques Monod and François Jacob, in Nobel Prize-winning work, by the way, showed how it worked for one particular process in the bacterium E. coli. And this was this was the system that they studied. And I won't go into it in any detail, except, the basic principle here is that this green protein here, produced by one gene, is able to bind to a site on DNA just before another gene, to bind to it and stop the transcription machinery from getting to that gene, and in that way it can turn the gene on or off. And this is quite an easy sort of idea to follow. It's basically just like a digital switch for turning for one gene turning another gene on or off. Now the problem is that gene regulation for large complex organisms like us, so-called metazoans, doesn't necessarily work in this nice, transparent, easy to follow way. It tends to involve a whole bunch of different components. Proteins called transcription factors, which are often actually another example of these intrinsically disordered proteins, and so a little bit promiscuous in what they bind to. There are some of these non-coding RNAs that I mentioned in fact, in general, they have roles in gene regulation. And then there are also other segments, other bits of DNA that are involved in this regulatory process. And some of those bits are a long way away from the genes that they're regulating. And so what has to happen is that there are these big loops of chromatin pulled out from the chromosome that have to be brought near to the gene that they're meant to be regulating. So there's this whole bunch of components that get together in a kind of loose, dense blob, a bit like a blob of vinegar in the oil of salad dressing. So, it looks like a total mess. So for us, gene regulation seems to be done by these, I like to think of them as loose committees of molecules, all talking to one another, sometimes rather indiscriminately. And it's really pretty amazing that somehow all of these components combine to make a reliable decision about whether to switch gene expression up or down, or on or off, despite the fuzziness of the interactions. And blobs rather like this, made from many different molecular components, seem actually to be involved not just in gene regulation, but in all kinds of cell processes, such as how cells deal with stress, such as heat, how they repair damaged DNA, and many others. And typically they're made up from proteins, and many of those are these intrinsically disordered proteins, and RNA. And in general, this non-coding RNA that doesn't encode proteins, that come together in these structures now generically called biomolecular condensates. And they seem to be an intrinsic feature of how cells organize and compartmentalize their molecular components. But it's a bit of a mess. It's a very fuzzy kind of logic that's going on here. And this sort of fuzziness, and the way that it produces collective decisions rather than simple digital logic based on precise molecular unions, this seems to be a characteristic feature of our molecular biology. Something like it actually is going on in another kind of non-coding RNA called microRNAs, which also intervene in and regulate genes. And it's these microRNAs were what won this year's Nobel Prize in medicine or physiology. We don't really understand how this logic works in general, but I think we can start to see why it is that for organisms like us, life does work in this kind of fuzzy, analog, and rather open-ended way. Because once you start to think about it, the more complex an organism is, the more the idea of a blueprint or a genetic instruction book for how it works starts to look like a really terrible way of designing an organism. If the organism working correctly depends on each of those instructions being executed perfectly at the right time and place, and every molecule being where it's meant to be when it's needed, then it's just not going to happen. It's a little bit like expecting some, incredibly complex piece of clockwork machinery to work, where each of these cogs has to be in exactly the right place. And in fact, it's even worse than that. It's like expecting this sort of mechanism to work when you put it in a warm bath and shake it around a lot, because that's what the molecular world is like. So rather than rely on a mechanism like that, we have these fuzzy molecular mechanisms where often the details don't matter. The committees can still come to good decisions even if some of their members aren't quite working properly or aren't there at all. And let me give you an example of how the body uses higher level principles like this that help organize our tissues and bodies and disperse the responsibility for that process rather than relying on this molecule doing exactly the right thing to that molecule. So we generally all have 5 fingers, right, and 5 toes. But there's no gene that specifies that. The way our fingers are now thought to grow is that we start off as embryos with little limb buds with sort of paddle-like endings with no fingers at all. And then a small set of developmental proteins interact with one another in a way that patterns them. And it patterns them in a way that was first predicted by the British mathematician Alan Turing in 1952. So Turing showed how it's possible for a soup of reacting chemicals to produce differences in concentration, even though all the molecules themselves are diffusing around, so-called Turing patterns. And one of the generic forms of these patterns is stripes. And it seems that our fingers grow as kind of radiating stripes in this limb bud, so that give rise to these differences in concentration where, where particular developmental proteins are concentrated, they trigger the development of the finger bones. And so the reason that there are just 5 of these is that the stripes have an intrinsic width, and they grow at just the right stage in development, where precisely 5 stripes will fit into the available space in that embryonic limb bud. And it seems that only a little tweak in the growth conditions or in the timing of growth can generate a different outcome, can generate more or fewer stripes. And that may be why much the same process can in other organisms, like in fish, can give rise to a greater number of stripes. And in fact, you also see that some of these stripes are bifurcating at the end, which is also exactly what the Turing mechanism would predict. So you see that here, the genes are they're not determining in any clear way from the genome itself what the outcome is. They're providing the resources, the basic resources that are needed for the body plan. You won't be able to read that body plan from the genome, no matter how much you stare at it. And the key genes and their product proteins involved in this process, they're not genes for making fingers. They're genes and they're proteins that turn up in a whole range of developmental processes. So they're kind of developmental resources that the developing embryo uses in various ways at various stages. So the story here isn't really about the genes themselves, but about how the cells and tissues are using them in just the right time and just the right place to create structure. And what these principles really amount to is taking the correct functioning for the building of the organism, or for the maintenance of the organism, off the lowest levels of the system, off the genes, and handing it to a higher level. In other words, the way we work seems very often to be designed to take the pressure off our genes, to make them, in general, no longer the real cause of our traits and behaviors, of our phenotype. And in that way, mistakes and malfunctions at the lowest level, say if you have a genetic mutation, can often be compensated for at a higher level of the developmental process. And the whole system then isn't just reading out instructions in a sort of automatic manner. It is also acting on information that it gathers at other levels, and sometimes information that comes from the surrounding tissues, and that can actually feed back to control what the genes themselves are going to be doing. And so, you know, Crick's central dogma is fine as far as sequence information is going, but if we're thinking of the more general information ecology of the organism, there's a much wider pattern, and in fact, it's an open informational system. And so what we have is really an emergence of causation at higher levels of this complex system. And when that happens, this is something that's very familiar to people studying complex systems, and they often talk about it as causal emergence. And it's like what we see in flocks of starlings, where the overall behavior of this system can't be ascribed to and isn't dependent on what any individual bird is doing. It is robust against one or two birds doing something eccentric in this flock. And we can, and in fact, this isn't just a sort of a loose metaphor. Causal emergence, there are various ways that you can quantify it, and it has been quantified in various organisms. And it turns out that we can see that there is more of it going on in organisms like us than there is in bacteria. Well, perhaps the ultimate expression of this causal emergence is our brains. You see, obviously, simple organisms like bacteria don't have brains, and they don't need them because they have a simpler life. The challenges they face are, really aren't that diverse. They have to find food, they have to find moisture, they have to figure out how to move to get there. And if they move outside of the environments that they tend to thrive in, then they die pretty quickly. But we're not like that. We get everywhere, and every day we face decisions and challenges that we have never faced before in our lives and our ancestors have never faced before. And so we cannot rely on our genome to tell us what to do in all of those circumstances. In the face of such unpredictability, you need some kind of higher level system to take the strain. And that's what the brain does. And the brain does it not by carrying out some precise computation of inputs and outputs, but by using, again, fuzzy rules of thumb that tell us, you know, this is probably the right thing to do. I'm not sure, but let's give it a try. That's what cells do as well a lot of the time. And this way of working is different from any kind of machine we have ever invented. So living organisms are not just kind of complicated soft machines. And what really distinguishes all living things from machines, or any machines that we've yet made, is that they're not automata that just respond in a stimulus-response manner. They have agency. What I mean by that is that they're able to manipulate and alter themselves and their environment in order to try to attain some self-determined goals. And we usually think of those goals as being survival and reproduction. But actually, you know, for all of us, we have plenty of goals that aren't obviously related to that at all. Your goal in coming here today probably wasn't clearly related to either of those things. And if it was, you're probably going to be disappointed. But I think that the same is true even for single cells, that at some level, even single-celled organisms have an agency that allows them to set their own agendas and not to be totally predictable. And I think that displaying agency is actually a much more fruitful and general way to think about what living systems are than to compile some checklist of criteria that they have to satisfy, like being able to reproduce and metabolize and homeostasis and so on. And that's why I think to have an overarching view of how life works, I think biology needs to develop an understanding of, and hopefully a theory of, agency. Now, there are practical reasons, of course, to want to understand how life works, not least because, as I implied at the beginning, sometimes it doesn't work quite as we want it to, and we have to try to figure out how to put it right. And that's the business of medicine. And if we want to bring about some change in a system, then we'll do best to intervene at the place where that outcome is genuinely caused. So we need to ask, is the cause of such and such a disease actually in the genes? And therefore we should be targeting the genes. You know, what we tend to hear about when in discussions of gene-based medicine are actually the exceptional cases where cause really does seem to be situated largely in a given identified gene. So we hear about conditions like like sickle cell disease, which does seem to be essentially a monogenic disease caused by mutations to a single gene. And so in that case, it is possible, and now it really is possible, to go in and intervene at that genetic level using a gene editing method like CRISPR to put it right. But most diseases aren't like that. Most diseases, when they're, the genetic component of them seems to be related to many, perhaps even hundreds or even thousands of genes, each of which has a tiny, tiny role to play. And in fact, it's even worse than that, because many of the linkages that are found between genomic sequences and diseases turn out to be in parts of the genome that don't encode proteins at all. They're in those non-coding parts of the genome, probably playing some regulatory role. So it's very hard to make interventions, probably impossible to make interventions at the genetic level, because ultimately that's not where causation lies. And that might be, it probably is, one of the reasons why genetic-based approaches to cancer treatments in particular have tended to be rather disappointing. You know, the idea, we know that cancers do arise or can arise because of mutations that happen to our genes, perhaps because we inherited one, or perhaps because we were exposed to something in our environment that caused it. But and so this has, in the past, led to the idea that we'll be able to find cures by looking at the genetic roots of cancer. But that, as I say, has been quite disappointing. And it turns out that often the most effective level of intervention is a higher one. Often, and this is often the case with all disease, often at the level of the immune system. We heard yesterday about the Keytruda anti-cancer drug, which is a drug that does its business by intervening with the in the immune system and helping the immune system to attack cancer cells. So this is how the cancer biologist Michael Yaffe put it in 2013. He said, "We've spent fruitless years looking for cancer-linked genes, not because we knew that they were the key to new treatments, but because we had the techniques for looking for them." So he said, like data junkies, we continue to look to genome sequencing when the really clinically useful information may lie someplace else. But quite aside from practical concerns like this, I think we should be interested in the question of how life works, simply to make us appreciate all the more how astonishing life is. The only way you can pretend that that question is going to be explained by genes themselves, it seems to me, is to end up making the genes like little agents, like little creatures all competing with each other. And that's not what genes do. That is not what genes are. So this idea that an account of life is to be found by gradually increasing the magnification until we arrive at molecules, this is probably heretical at a Molecular Frontiers meeting to say this, but this is what the Nobel laureate biochemist Albert St. Georgi said. He said, "My own scientific career was a descent from higher to lower dimension, led by a desire to understand life. I went from animals to cells, from cells to bacteria, from bacteria to molecules. On my way, life ran out between my fingers." So what it comes down to then is looking for an explanation of how life works that really does justice to the majesty, the truly amazing nature of life itself, saying that life is a machine made by genes or like a computer running a program is not only wrong from the perspective of what modern biology tells us, it seems to me to banish biology, to banish life altogether from the question. The eminent physicist Michael Berry told me recently that he was once asked by an interviewer, what is the biggest unsolved problem in physics? And he imagined that the questioner was expecting him to give some standard answer like dark matter or quantum gravity, but he found himself saying that if, as we think, all matter is described by quantum mechanics, where does the aliveness of living matter come from? Now, he didn't mean that it must come from some quantum explanation. He meant that living matter seems to be, and in fact is, profoundly different from other kinds of matter, and we don't really know why. Parts of the universe, all of these parts, have become aware of themselves and their place within it. And I'd like to humbly suggest that biologists should never forget that what they're really trying to do, to explain and to understand, is that. So no wonder it's really hard. But I do believe it's possible, and I do believe also that we're in one of the most exciting periods of that exploration. So for people who are thinking of starting a career in biology, I personally think there's never been a more exciting time to do it than now. Thank you.